All right, hello, good morning, everybody. My name is Felix. I'm a postdoc at Stanford University, where I work in the lab of Manu Prakash. We're in the bioengineering department, and together with Hari Priya, we develop novel techniques for vector surveillance and mosquito ecology. I'll start, I guess, with a little apology that this slide at least is showing up all the way, so that's good. So I'll share with you one of the projects that we've been working on for a while. And I'll just have a very, very brief introduction to kind of motivate our work. And I think that, basically the audience here doesn't need that much introduction. So I'll keep it very brief. So this figure kind of motivates me a lot. On the left here, you see the proportion of 2 to 10-year-olds that has malaria in sub-Saharan Africa. And you see that we have very high resolution data on that. So on the human cases, we have very high resolution data. Also, we have very high resolution data in time. So that's great, but if you then look at the mosquito data that is present, you see that there's large areas, for instance, this area over here, or in the center over here, where we have very, very few data on what type of mosquitoes are actually present over there. I'm sorry, I'll only be pointing on this screen. I guess the point that I'm trying to make is that decent maps of mosquito abundance are very, very scarce. There's not a whole lot of data on what kind of species are there, what subspecies are there, what are, for instance, the infection rates of those species with parasites or viruses. Do they have co-infections? There's very limited data on that. Oh, sorry about that. I'll just stand back a little bit. Okay, so this is a big challenge, and why is it? I guess the question is, why is this data so scarce? Well, one reason why that data is so scarce is that the current methods which people trap mosquitoes or kind of try to do mosquito ecology are, I've just put 2 examples over here. There's human landing catches, or there's, for instance, the CDC trap. There's also BG traps. There's all kinds of traps. And those traps usually use electricity. They use CO2. So they use things that are not typically available in the field that you have to bring yourself. Also, both landing catches and trapping is fairly labor intensive. And then with these methods, if you collected a lot of mosquitoes, then you were faced with a little tray like this, or even, yesterday we saw a talk where somebody had a gigantic bucket full of mosquitoes. So you actually have to process all those mosquitoes individually. So you have to sort them by their species, and then you have to PCR them or dissect them or something. So all these things are very labor intensive. One way of coping with making it a little bit less labor intensive and cheaper is to just pool a bunch of mosquitoes into one big pool and PCR that one, but that of course lowers the resolution, because if the pool is positive for, let's say, Zika, you don't know whether that was one mosquito or maybe all 100 mosquitoes in that pool. So what would be kind of an ideal trapping method? So we tend to think of that an ideal trapping method would have single mosquito accuracy, so it would be really nice if you could just do your data on a single mosquito level. That also, of course, necessitates high throughput, because in a lot of places, the infection rates of mosquitoes are really low. For malaria, for instance, I've read numbers as low as 1 in 1,000, so you need to actually look at a lot of mosquitoes before you find a positive one. And of course, it's nice when methods are genomics compatible, and cheap is always a good thing if you want to do stuff in high throughput. So we've been thinking about how to tackle some of these challenges at least, and we're not quite there yet, but we're moving towards this goal. And I'll share with you one project that we've been working on for about a year now, which is called the vector chip. And the basis of the vector chip is that mosquitoes are actually very good at pipetting. So mosquitoes have this little mouth over here. Oh, no. Well, actually, that's not that bad. So they have this mouth, the proboscis, which is about 60 micron in diameter. So it's actually a very fine pipette. And here you see a movie where a mosquito is salivating into a pipette tip, and you see these little droplets shooting out. And those little droplets actually, they contain everything we need to know about the mosquito. So we're trying to move away from using whole insects. We just only want to use the saliva, because the saliva holds viruses, it holds parasites, and we're trying to figure out whether it also holds enough mosquito DNA to actually do the species identification. So the platform that we're making, the vector chip, is basically, you can see it as a little business card size or maybe a bit larger chip. On this chip, we integrate a lot of cues, so for instance, odorants, temperature. You can also think of texture, color, all kinds of things we can integrate on this platform. And in that way, it mimics kind of human skin. And because it mimics human skin, mosquitoes like it. They fly towards it, they land on it, and then the idea is that they actually bite the chip. When they bite it, they deposit their saliva, which has all the biomarkers that we want to look at, it deposits the saliva in a little pocket, and well, then we do some analysis on that little pocket. So the idea is that we'll just do on-chip, for instance, multiplex PCR, and then in the end you'll get a readout in which, for instance, a pocket that was positive for the red dye and the green dye tells you that this was a certain Anopheles species that was carrying malaria parasite. So over the coming couple of minutes, I'll break down the project in little pieces, and I'll go through, well, basically what those pieces are and where we stand at this moment in developing this project. So I'll first talk a little bit about how to attract mosquitoes to, well, basically a small surface, and whether we can actually verify whether they bite that surface or not. And after that, I'll very, very briefly go into how we analyze currently the samples that are on that surface. So I'll mainly be talking about, or actually only talking about, laboratory experiments. So what we typically do is that we have a cage of mosquitoes. Most of the data that I'll be talking about is Culex tarsalis. We've done some work on Aedes aegypti as well, and we have them sitting in a cage. We have a couple of cameras that are looking at that cage, and here, for instance, you see. So we do everything. So Culex tarsalis is a night biter, so we do everything in the dark with only red light. Mosquitoes can see red light, so for them it's dark. And we basically just present a little device on one face of the wall. We image it with one camera from, let's say, the right to get the population statistics on what happens on that entire wall. And then often we also have a higher resolution camera from the other side where we can actually see the mosquitoes biting the device. So at the moment we're benchmarking, did it advance? No. At the moment, we're benchmarking our assay. So we just do a couple of little tests of which we already kind of know the outcome. So we present 3 devices on the wall of the cage, in which all 3 devices are just a piece of agar hydrogel that have parafilm wrapped over it, and the parafilm is rubbed on somebody's skin such that it becomes a little bit more attractive. And we have 2 of those devices that we keep at 37 degrees. So that's the top one and the right bottom one. So it's at body temperature, and the left one over here is just at room temperature. And not very surprisingly, the mosquitoes really like. So here on the top right, I'm showing you every single dot there is a mosquito landing over about 4 hours. And of course, the mosquitoes really like the warm areas, and they don't really like the cold area. Over here, you can see basically what the statistics over time look. So in magenta, you just see what the baseline is. So that's just mosquitoes landing on the wall. And then you see that the 2 warm devices, so that's the blue and the red line over here, they get a lot of landings. And after about 1 or 2 hours, the activity peters off a little bit. So we can do this with a lot of different devices. So over here, we, for instance, have one that has just a hydrogel without any parafilm over it. So this is a very wet surface. This is a warm surface with parafilm on it. This is a cold surface with parafilm on it. So these 2 are, again, the same as on the previous slide. And what you'll see is that we get a lot of landings on the device on the right. On the left device, we, again, don't get a lot of landings because it's cold. But actually, on the top device, we get a lot of landings at the perimeter of the device. So this surprised us a little bit because this was actually agar with sucrose in it, so there's a food source for the mosquitoes there. We thought they were going to like it, but actually it's wet and it's presented in a vertical direction, so maybe it's harder for the mosquitoes to land on a wet surface that is vertical. So I guess and we can also, of course, track the mosquitoes. We can see where their activity is highest. We can see actually that, for instance, on this top device, they mainly land on the outside, then walk up a little bit, and then decide it's not such a good idea and go back again. So I'm showing you to give you a couple of basically to give you a sense of what we do, and we're now in a process where we're testing a whole bunch of different odorants that we can integrate on these devices, different colors, lures. So we're basically doing a broad screen and just trying to figure out what maximizes landings on the device. Well, so if we have landings, then of course after that we like to see whether they bite. So initially we were working a lot with PDMS because PDMS is a very nice material for us to work with. It's very easy to microfabricate it. But it actually turned out that the mosquitoes found it very hard to bite through. So you see them biting over here sometimes, but we never saw the proboscis actually go through. So that's when we switched to these agar and parafilm devices. And there we actually saw, so I'm showing you here a couple of videos. Don't seem to be playing. All right. sorry about that. So let's just stick to this picture over here. So over here you see a mosquito that landed on such a device. And this is the sheet of the proboscis. And you can actually see that the sheet of the proboscis normally wraps around basically the food kennel. So when that goes into a surface, the sheet folds back. And the fact that you get this fold over here means that the proboscis actually went into the device. So when we saw this, we got kind of happy. So because we knew that we made something that mosquitoes like enough to actually even start probing onto and biting into. So then we basically came up with the next assay where we have an acrylic piece that has little holes drilled into it. And in those little holes, we put agar gels. And then we had basically the same assay, but we're now filming it from the back so that we can have very high resolution imaging. And we see that mosquitoes really dig into the holes. And over here I'm showing you basically a zoomed in version where you really see the proboscis of the mosquito going into that tiny little hole over there. So of course the next thing that we now need to do is to analyze what the mosquito actually deposited into that hole. We're not quite there yet, unfortunately, so the analysis is something where we're still developing a lot of our tools at the moment. So we're a lab that does a lot of microfluidics. So over here, for instance, was a recent paper from our lab where we're just playing around with routing around droplets. This is something that we might in the end do with the saliva droplets of the mosquitoes. But for the moment, we just have these acrylic parts where we have hydrogels sitting in these little holes. Those hydrogels are pre-filled with PCR master mix. So this is actually a 100 nanoliter well that has PCR master mix in it. And what we've done on this figure over here to just demonstrate a proof of concept is that with a pipette tip, we've added a tiny little bit of mosquito DNA, and we can actually see that we can just run the PCR on this device, and we get positive results where we need positive results and negative results where we didn't put any DNA. So it shows you that the on-chip PCR at least is working. So where we're going from here, so we basically have a functional attraction assay. So we have an assay with which we can just quantitatively estimate how attractive chips are. So we're going to do a broad screen of a lot of cues. We also have the on-chip PCR working, so we're going to now do this with actual saliva samples. And because we want to maximize the information that we can get from saliva, we are also going to do a broad screen of saliva where we can figure out what DNA and RNA actually is in saliva. So with that, I would like to acknowledge our funding sources and collaborators, and I'd be happy to take any questions.
Thank you, Felix. We have a sliver of time for maybe a couple of questions. Yes.
Sorry, Natasha from the London School. I was wondering, how do you see that can be used on the field? Like, because we want to analyze the mosquitoes, we want to know if they're exposed or infectious species, where they have been studied.
Yeah. Sure. So in the field, we intend to do a bunch of things. So we're in places where there's lots of mosquitoes, we'll just expose these. So we've been trying this in Madagascar last month. We just expose these chips, and then we take them back to the field for analysis. So we don't see it as something that only works in the field. We will expose the chips somewhere in the field, then we take them back, and we just run the PCR in a lab. Of course, there's ways around that where you can, for instance, use isothermal assays to run your PCR, and then you can try to make something that is field compatible, but that doesn't have our interest right now. In addition, you could, of course, also, if there's places where, so we try to make the chip as attractive as possible to mosquitoes, but of course, it's really hard to beat like an actual human or a very large trap that has a lot of electricity and other stuff going into it. So you could also imagine that we just place this device inside a trap, so then you still have the problem that you need to use the trap, but at least we can get the individual mosquito bites, extract those out and PCR those. So then you would only have basically on my challenges slide, you would only have the first challenge of having the need for a trap, but the processing would be much easier.